What a great research.
Right.
Our next presenter is Ashley Hughes, and she will be presenting Smoking Cessation, Saving Lives, one
CYP2A6.
CYP2A6 at a time. Sure what that is.
Well, we'll get to that. As Dr. Snyder said, I'm Ashley Hughes. I'm on my third year here at Eastern, and for the last year I've been working with Dr. Denton in his research lab. And like he said, my project is smoking cessation, saving lives, one CYP2A6 at a time. So first off, we're just going to talk a little bit about smoking. So smoking is one of the leading causes of preventable death in the world. By statistics that have shown, over about 500,000 people a year die in the United States from smoking-related causes. And these numbers is probably a gross understatement of the true value. This is just what science is willing to categorize as smoking-related deaths. It can cause diseases such as lung cancer and chronic obstructive pulmonary disease, stroke, emphysema, breathing disorders, and so many others. And every single year, billions of dollars are spent not only to inform people of the hazards of smoking, but also because of the health problems that it causes. So statistics have shown that about 21% of adults are smokers, and the majority of these people that smoke express a desire to stop smoking. However, even though most of them want to quit, less than 5% are successful if they're trying to do this unaided. And even if they are using some form of aid, the success rate is still very low. And statistics also show that 70 to 80% of these people are going to relapse in the first 12 months of trying to quit smoking. So right now, there's a couple different options for people who want help in quitting smoking. The most well-known one is nicotine replacement therapies, such as Nicoderm or Nicorette, which is using patch or gum to replace the nicotine in your body so you can try to not smoke. And there's also antidepressant drugs and inhibitor drugs that are out there that can be prescribed also that have different effects. So nicotine is the chemical that is responsible for the addictive nature of smoking. Nicotine triggers the release of dopamine, which makes a feel-good sensation. So people smoke, they feel good, they want to maintain that feel-good sensation by maintaining a semi-constant level of nicotine in their bodies. Nicotine is metabolized into cotinine mostly by an enzyme, cytochrome P450-2A6, or CYP2A6. There we go, that's where that came in. And there's a couple factors that affect how this enzyme metabolizes it, such as diet, gender, their status, if they're a smoker or not. But the main one that I'm going to focus on is genetics. And in the corner there is a crystal structure of the CYP2A6 enzyme. So there's a lot of genetic variations in the gene that codes for the CYP2A6 enzyme. And certain variations that have been studied are shown to decrease the activity of the enzyme. So different populations have different percentages of this, the variations that appear in their populations. And when the activity is decreased, the metabolism of nicotine is also decreased. So what does this mean for those individuals that have this genetic variation? It means that the nicotine stays in their body longer, so they have higher prolonged levels of nicotine with the same amount of smoking as someone who doesn't have the deficient genes. So this means that they smoke less frequently, that they have a desire to smoke less because the nicotine stays in their body longer, and so their overall consumption of nicotine is less than those who have a normal functioning CYP gene. So the goal of my research is to mimic those genetic slow metabolizers with competitive inhibitor drugs. So the drugs go in and they bind to the enzyme so that nicotine is competing for that space and nicotine isn't metabolized as quickly. So from what we know about the enzyme, the desired structure that we're looking for is a pyridine ring with a linker group, which is just to fill space in the enzyme, and a basic nitrogen. So we've gotten crystal structures of the enzyme and some of the compounds that we have made. And so what we know about it and how it binds is that the basic nitrogen is coordinating with the ion of the heme group in the enzyme. And the nitrogen on the pyridine ring is hydrogen bonding with a specific residue in the enzyme, number 297, which is an asparagine residue, and it's forming hydrogen bonds that hold in place. There's also aromatic interactions with some of the other residues that are part of the enzyme that keep and hold it in place. So this is kind of what has led us to the basic structure of what we're aiming to have our structures of the compounds I synthesize look like. So now onto my specific stuff, the chemistry side of it. I've made a procedure to synthesize a set of compounds. For me, I have my pyridine ring, my linkers, and isoxazoline ring, and then I have my basic nitrogens on the end. So what I do is I start with the aldoxenes. So I have my pyridine ring with an aldoxene. I then chlorinate it with N-chlorosexcinnamide to make the first intermediate. From then I do a 3+2 cycle addition with my various propargylamines. So the two that I mostly use are N-dimethyl propargylamine and N-Bach propargylamine. After the cyclization happens, on the N-Bach I have to do a further step to deprotect it to get the primary amine, because our research has shown that with the N-Bach group still attached, it doesn't fit. It's too big to get into the enzyme really. Once I've made my compounds, then I have to purify them and then I can send them off for testing. So once I've figured out my reaction mechanisms and scheme, I focused a lot of my work on optimizing the reaction, so getting it to do better, faster, more efficient, greener products. So I'm not going to focus too much on the mechanism, so I don't want to overwhelm people, but this is what we have proposed that how the reactions proceed. The chlorination with N-chlorosexcinnamide is a radical reaction with the aldoxane. And then here's the proposed mechanism of that 3+2 cycle addition with the propargylamines. So the end goal. The end long-term goal of my research is to synthesize a library of compounds that can be sent off and tested as potential drug candidates. And these drug candidates would be used alongside the nicotine replacement therapies to help increase the odds that people would stop smoking. So nicotine replacement therapies right now haven't shown a great percentage of success in helping people stop smoking, but using it alongside this type of drug that we're hoping to make, it would hopefully increase those percentages and help people stop smoking. So on the screen, some of the compounds that I've either made or in the process of making, and you can see that they all have the same basic structure, with the pyridine ring, isoxazole, and nitrogens, and they're kind of just different in the substituents that are on them. And the substituents kind of can just also have other forms of interactions, hopefully with the enzyme, that might increase the binding efficiency of these compounds. I just wanted to thank Dr. Denton for all of his help and research in the lab, and ask if you guys have any questions. Josh.
How's the purification run? How do you guys purify it?
Yeah, cool. So after I synthesize my compounds, I do an extraction first of all. I've actually been able to get an extraction procedure that has made it very efficient. For the most part, after the extraction, which I use 1 molar sodium hydroxide and diethyl ether. And with that, my compounds are almost pure at that point, which is really awesome because it took me a long time to figure this out. After that, though, just to get the final purification, make it really good, is I usually run columns and just have to find a solvent system that works so that the RF value is low enough. Yes?
If any of your compounds actually became drugs that people could take, did you think about maybe what possible side effects there could be of people taking it?
I haven't really thought about that on like the biology side of it, but we also know that some of the other compounds that are similar to what I make, that's research has shown that they don't bind with many of the other enzymes in the liver, because that would be a problem if they bind with my enzyme, but they also bind with a lot of the other ones, that they could be cross reactions that could affect the body. But because the studies have shown that my style of compounds are only really binding with the CYP2A6 enzyme, and they're not binding with the other liver enzymes, and they've also shown that they're not binding with the nicotinic receptors in the brain, that there decrease the possibility of those being those bad side effects.
Just this is not a chemistry question, but in the slide where you showed the different groups that had genetic predisposition to be impacted, I was trying to make sense of that. Which groups were the ones who were most reacting to cigarettes?
Oh, okay. Yeah, let me
just.
Okay, so what it's showing is that the white is the people that have the normal CYP2A6 activity. So their body metabolizes nicotine at the normal rate. When we get into the gray and the dark gray and the black, those are showing decreased activity. So when we get into this dark colors, so we're in the Chinese, Korean, and Japanese populations, they are metabolizing nicotine slower. So studies have shown that they're less likely to become addicted to nicotine because their body, like they can metabolize it slower and they don't feel that urge to smoke as often. You're welcome. Yes?
On that last slide, previous to the credit slide,
Okay.
you showed a bunch of molecules that you want to make or have made. Which
Yes.
ones have you made and how do you characterize them?
Sure. So the ones that I have made just the basic unsubstituted pyridine ring, the indole, the 2-methoxy, the 6-trifluoromethyl, and the 6-bromo. And I've also been able to use our LCMS and also NMRs to characterize what I have.
Who's NMR?
Oh, actually, no. So this is really cool. So our NMR's been broken or been down for a while, but we've been able to be lucky enough to let Gonzaga has been letting us go use their NMR. So driving close enough, we can go use their stuff and come back.
Have you received any results on binding affinities with those compounds in the enzyme?
Yeah, I've gotten a few back. For what I've gotten so far, nothing super great, but it's even bad data or data that doesn't look great. It can lead you on to the good, like new stuff. So I know, oh, this didn't work. Let's change it and see if this works better, and then you can get ideas of things that might be new compounds that I want to make.
Okay. Well, let's thank Ashley.